Hello, welcome you all to the biology class. Today we are here in the 13th lecture of our biomolecules. Today we will discuss about the factors affecting the enzyme activity. Okay, yesterday we have made a brief list of those factors like temperature, pH, concentration of the substrate and the presence of inhibitor, water, light, etc. or some radiation. Okay, but as of our text we will discuss 4 major factors that affect the enzyme activity. It means any factor, if it is in a more concentration, that may affect the enzyme activity. If any factor is in less concentration, that may affect the enzyme activity. Like that, there are many factors which will affect the enzyme activity in the living systems, in the living organisms. Okay, among them we will discuss the most important one and those which are mentioned in our text. Right, the first factor that affect the enzyme activity is temperature. Better we will study temperature and pH together only, that's good. So temperature and pH. Okay, so both have a similar effect on enzyme activity. Okay, so what happens here is, whenever the temperature, pH is increasing, the enzyme activity will also increase. Enzyme activity will also increase, but only up to certain limit. After that enzyme activity will be declined. ಅಂದರೆ ಈ ಟೆಂಪರೇಚರ್ ಮತ್ತು ಪಿ ಹೆಚ್ ಒಂದು ಮಟ್ಟಿಗೆ ಇನ್ಕ್ರೀಸ್ ಆಗುವವರೆಗೆ ಎಂಜೈಮ್ ಆ್ಯಕ್ಟಿವಿಟಿ ಜಾಸ್ತಿ ಆಗುತ್ತೆ, ಮತ್ತು ಆ ಲಿಮಿಟ್ ಕ್ರಾಸ್ ಆದಮೇಲೆ ಮತ್ತು ಎಂಜೈಮ್ ಆ್ಯಕ್ಟಿವಿಟಿ ಡಿಕ್ರೀಸ್ ಆಗುತ್ತೆ. ಓಕೆ, ಫಾರ್ ದ್ಯಾಟ್ ದೇರ್ ಇಸ್ ಅ ವರ್ಡ್ ಕಾಲ್ಡ್ ಆಪ್ಟಿಮಮ್ ಟೆಂಪರೇಚರ್ ಆ್ಯಂಡ್ ಆಪ್ಟಿಮಮ್ ಪಿ ಹೆಚ್. Optimum temperature or the optimum pH. Okay, for example, when we say temperature, the 37 degrees Celsius normal temperature where the reactions occur normally. Okay, when we come to pH, one example is a pepsin enzyme, which will help in the digestion of the proteins. We will discuss in human physiology. So this enzyme will be active at the pH of 1.4. Like that, for every enzyme there is an optimum temperature and there is an optimum pH. Optimum temperature and optimum pH. Okay, better we can show with the help of a graph. So when we plot a graph by taking the temperature on x-axis and enzyme activity on y-axis. Enzyme activity on y-axis. Okay, so as temperature increases, the enzyme activity increases. Okay, so initially as the temperature keep on increasing, the enzyme activity will also keep on increasing, but after certain range, when temperature has reached its limit, the enzyme activity will also becomes normal and it will slowly starts decreasing. ಓಕೆ, ಮೊದಲಿಗೆ ಟೆಂಪ್ರೇಚರ್ ಜಾಸ್ತಿ ಆದಂಗೆ ಈ ಎಂಜೈಮ್ ಆ್ಯಕ್ಟಿವಿಟಿ ಜಾಸ್ತಿ ಆಗ್ತಾ ಹೋಗುತ್ತೆ. ಅದೇ ರೀತಿ ಆಪ್ಟಿಮಮ್ ರೇಂಜ್ ರೀಚ್ ಆದಮೇಲೆ, ನಾರ್ಮಲ್ ರೇಂಜ್ ಲಿಮಿಟ್ ಕ್ರಾಸ್ ಆದಮೇಲೆ ಆ ಎಂಜೈಮ್ ಆ್ಯಕ್ಟಿವಿಟಿ ಕಡಿಮೆ ಆಗ್ತಾ ಬರುತ್ತೆ. ದಿಸ್ ಈಸ್ ಹೌ ದ ಟೆಂಪ್ರೇಚರ್ ಹ್ಯಾಸ್ ಎಫೆಕ್ಟ್ ಆನ್ ದ ಎಂಜೈಮ್ ಆ್ಯಕ್ಟಿವಿಟಿ. ಸಿಮಿಲರ್ಲಿ ವಿತ್ ಪಿ ಹೆಚ್ ಆಲ್ಸೋ, ವೆನ್ ವಿ ಪ್ಲಾಟ್ ಅ ಡಯಾಗ್ರಾಮ್ ಆಫ್ ಪಿ ಹೆಚ್ ಆ್ಯಂಡ್ ಎಂಜೈಮ್ ಆ್ಯಕ್ಟಿವಿಟಿ, ಸೊ ಪಿ ಹೆಚ್ on x-axis and enzyme activity on the y-axis. So initially as the pH increases, the enzyme activity will also increase, but as soon as the pH reaches the limit, means the enzyme reaches the pH limit, then the activity will also be reduced. Activity will also be reduced. Okay, you will commonly say in Kannada, ಅತಿ ಆದರೆ ಅಮೃತ ಕೊಡೋ ವಿಷಯ ಅಂತ ಹೇಳ್ತೀರಿ. Like that, every factor has a limit. Every factor has a limit. It may be temperature, it may be pH, it may be water concentration, it may be the concentration of substrate, maybe concentration of enzyme. Any factor which has a limit. After crossing that limit, the enzyme activity will also get reduced. Okay. One more thing, when we come to temperature, it has various effects. Means one type of effect in high temperature, one type of effect in low temperature. Okay. So at high temperature, what happens? The enzyme gets damaged. Just because enzyme is made of protein is made up of amino acids combined by peptide bond. So it is high temperature, the peptide bond will be broken down so that the denaturation occurs. Okay, it is just because of the denaturation. Okay, but at low temperature, what happens? At low temperature, here the enzyme becomes inactive. Okay, the enzyme will not be destroyed, but the enzyme becomes inactive. Enzyme will be preserved at inactive state. Inactive means enzyme will lose its activity, but enzyme will not lose the nature. ಓಕೆ, ಎಂಜೈಮ್ ಹಾಳಾಗೋದಿಲ್ಲ, ಆದರೆ ಅದರ ಆ್ಯಕ್ಟಿವಿಟಿನ ಕಳ್ಕೊಳ್ಳುತ್ತೆ ಅಷ್ಟು, ಅದು ಕೆಲಸ ಮಾಡಲಿಕ್ಕೆ ಆಗೋದಿಲ್ಲ. ಓಕೆ, ದಟ್ಸ್ ಹೌ ದ ಬೋತ್ ಪಿ ಹೆಚ್ ಆ್ಯಂಡ್ ಟೆಂಪರೇಚರ್ ಹ್ಯಾವ್ ಎಫೆಕ್ಟ್ಸ್ ಆನ್ enzyme activity. Initially increase when temperature increases, pH increases, and when temperature reaches the limit, pH reaches the limit, the enzyme activity slowly declines towards the minimal value. Okay, that's how we can study the effect of temperature and pH on the enzyme activity. Okay, next we'll move to the next factor after this, after temperature and pH. Very important factor and very interesting factor, the second factor, that is concentration of substrate. Concentration of substrate. Okay, so imagine I will label substrate with these colors and enzyme with this color. Okay, so there are 10 substrate molecules. 4, 5, 6, 7, 8, 9, 10. There are 10 substrate molecules. Okay, and whenever there are only 6 enzyme molecules, only 6 enzyme molecules, so the process will be little faster. Okay, means the process keeps on increasing, enzyme activity keeps on increasing, and when it has almost all equal number of enzyme and substrate, it becomes normal. It becomes normal because it has reached saturation. Means the velocity has reached maximum because all the substrate molecules bind with the enzyme molecules. It has reached the saturation, equal number of enzymes, equal number of substrates. So maximum velocity will be observed in the enzyme activity. Okay, but afterwards, even if you increase the number of the enzyme molecules, there is no effect. Even if you increase the number of substrate molecules without enzymes, there is no effect, because they are free only. They are free only. Just the process becomes normal. The graph will be horizontal afterwards, because the activity will be increased only till reaching the saturation. Means equal number of substrates, equal number of enzymes. Okay, that is how the substrate affects. So we will draw a graph for that to understand better. Okay, it's very important graph for examination also. So observe carefully. So y-axis will take velocity of the reaction, x-axis will take substrate concentration of substrate on x-axis. Y-axis will take a velocity of reaction. We'll take it V, velocity of a reaction, right? So here we have a various concentration of substrate in the reaction, any container, and we can mark any levels or velocities of the reaction, any velocities of a reaction. Okay, as the concentration of the substrate increases, the velocity of the reaction also increases. Means the rate of reaction increases, enzyme activity increases. Okay, so it will be a slightly slope, moving in a forward direction, moving in a forward direction. I told you, when the number of substrates equals to number of enzymes, number of substrates equals to number of enzymes, what happens? It reaches the saturation. Means all substrate molecules are bound with the enzyme molecules. So this becomes a slope now. This becomes the slope now, right? So at this point the velocity is maximum. At this point the velocity will be maximum. Okay, this point, this velocity we can say it as a V maximum. We can call it as a V max. Means maximum velocity. When we come to this is the concentration of the substrate. This is the concentration of the substrate. At this concentration of the substrate, velocity is maximum. Okay, when we will take a half of this, almost it will come here. So measuring the half velocity and half substrate concentration, not half, this may be less than half even. So at this concentration of the substrate, velocity is maximum. We call it as Vmax. At this concentration of the substrate, velocity is half. So this point, this we can say as Vmax by 2 or half Vmax. Means this divided by 2. Okay, and this substrate concentration is known as Km. This substrate concentration is known as Km. Okay, I'll tell you what is Km. Km, name of scientist, that is Michaelis-Menten constant. Michaelis-Menten constant. Okay. In the examination, you will get a question, one more question, what is Km or what is Michaelis-Menten constant? The answer would be the concentration of the substrate at which velocity is half maximal. Concentration of the substrate at which the velocity of the reaction is half maximal. Better I will write it here. The concentration of substrate at which the velocity of reaction is half maximal or half maximum. Okay, this is our definition of Km. Definition of Km. Okay, means concentration of the substrate at which the velocity of the reaction is half maximal. This is maximal. This is a half maximal. Okay, and you should also remember this is reaches the maximum limit when it reaches the saturation. This is the saturation point. Saturation point, right? So this is how the concentration of the substrate affects the rate of a reaction. Okay, actually it is just analysis of the concentration. Okay, we can just say as concentration of the substrate increases, velocity increases. When it reaches the saturation, the graph becomes horizontal. But when we analyze with the help of a graph, we can get the Km, Michaelis-Menten constant, that is concentration of the substrate at which the velocity of the reaction is half maximal, half of the maximum. Okay, so remember this. You may get even 5 mark question on this. Okay, or a 2 mark question just for the graph, or 1 mark question for this answer. What is Km? Okay, what is Michaelis? Don't write only this much. It's not the answer. The answer should be this. If this is asked, how to write this, then you should write this. Okay, mention what is Michaelis, what is Km, and what do you mean by Km. Okay, that's how the concentration of the substrate affects the rate of a reaction. Right, this is the second and very important factor for your enzyme action. Okay, moving on to the third factor. And very interesting factor. First we will write that factor here. So the second factor was concentration of the substrate. Concentration of substrate. Third factor is the presence of inhibitors. Presence of an inhibitor. So this we have discussed. So finished. We'll now discuss about the third factor, the presence of an inhibitor. Okay. So here the question is, what is inhibitor? Okay. Imagine we have a home. Mani ada. ಮನೆಗೊಂದು ಡೋರ್ ಅದ, ರೈಟ್? ಡೋರ್ ಇದಿರಲೇಬೇಕಲ್ಲ. ಸೊ ವಿ ಹ್ಯಾವ್ ಅ ಡೋರ್ ಆ್ಯಂಡ್ ವಿ ಹ್ಯಾವ್ ಅ ಲಾಕ್ ಹಿಯರ್. ವಿ ಹ್ಯಾವ್ ಅ ಲಾಕ್. ಮನೆಗೊಂದು ಲಾಕ್ ಹಾಕಿರ್ತೀರಿ, ರೈಟ್? ಈಗ ನೀವು ವಿಲ್ ಜಸ್ಟ್ ಎನ್ಲಾರ್ಜ್ ದಿಸ್ ಲಾಕ್ ಫಾರ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡಿಂಗ್ ಬೆಟರ್. ದಿಸ್ ಈಸ್ ಅವರ್ ಲಾಕ್ ಆಫ್ ಅವರ್ ಹೋಮ್. ನಮ್ಮ ಮನೆಯಾಗ ಲಾಕ್ ಹಿಂಗಿಲ್ಲ ಅಂತ ಕೇಳಬೇಡ್ರಿ. ಲಾಕ್, just like lock. Okay, the pattern of this lock is like this. So you have a key like this, then only the lock opens. ಈ ರೀತಿ ಇದ್ದಾಗ ಮಾತ್ರ ಲಾಕ್ ಓಪನ್ ಆಗುತ್ತೆ. ಓಕೆ, ವಿಲ್ ಜಸ್ಟ್ ಡ್ರಾ ಒನ್ ಲಾಕ್ ಹಿಯರ್, ಕೀ ಹಿಯರ್. ಸೊ ಇಟ್ ಮಸ್ಟ್ ಬಿ ಲೈಕ್ ದಿಸ್. Okay, this must be the key. Okay, so this is the lock, this is the key, right? Then only the lock opens, right? But ಮನೆಗೊಬ್ಬ ಕಳ್ಳ ಬಂದ ಬಂದು ಲಾಕ್ ತೆಗಿಲಿಕ್ಕೆ ಟ್ರೈ ಮಾಡ್ದ. ಹೆಂಗೆ ತೆಗಿತಾನ? ಹಿ ವಿಲ್ ಬ್ರಿಂಗ್ ಅನದರ್ ಲಾಕ್ ಆಫ್ ಹೀಸ್ ಓನ್ ಪ್ಯಾಟರ್ನ್. ಓಕೆ, ಸೊ okay, so this is a fake key. Okay, original lock, original key, but the fake key. But this key can go inside but cannot open the lock. ಈ ಕೀ ಒಳಗೆ ಹೋಗ್ಬೋದು, ಆದರೆ ಲಾಕ್ ಓಪನ್ ಮಾಡಲಿಕ್ಕಾಗಲ್ಲ, ಲಾಕನ್ನು ಓಪನ್ ಮಾಡಲಿಕ್ಕಾಗಲ್ಲ. ಯಾಕೆ? ದಿಸ್ ಪ್ಯಾಟರ್ನ್ ಈಸ್ ನಾಟ್ ದೇರ್, ದಿಸ್ ಪಾಯಿಂಟ್ ಈಸ್ ಮಿಸ್ಸಿಂಗ್ ಓವರ್ ಹಿಯರ್. ಈ ಪಾಯಿಂಟ್ ಮಿಸ್ ಆಗಿದೆ, because this point is also necessary to open the lock, right? So ಕಳ್ಳ ಬಂದು when he insert this and when it breaks down inside this, the lock will be blocked. ಕೀಯನ್ನು ಒಳಗೆ ಹಾಕಿದಾಗ ಅದು ಮುರಿದು ಹೋಯಿತು. ಓಕೆ, ವಿಲ್ ಜಸ್ಟ್ ಪುಟ್ ದಿಸ್ ಇನ್ಸೈಡ್. ಸೊ ಇಲ್ಲಿ ಬಂದಾಗ ಕೀ ಮುರಿದು ಹೋಯಿತು. ಆವಾಗ ನಿಮ್ಮ ಕೀಯನ್ನು ಹಾಕಲಿಕ್ಕಾಗಲ್ಲ, ಲಾಕ್ ಓಪನ್ ಮಾಡಲಿಕ್ಕಾಗಲ್ಲ, ಹೌದಾ? ರೈಟ್? ಸೊ ನಾವು ವಿ ಕ್ಯಾನ್ ಸೇ ದಿಸ್ ಈಸ್ ದ substrate, this is the substrate, this is the enzyme, and this is the inhibitor. ಅಂದ್ರೇನು? ಈ ಫೇಕ್ ಕೀ ಇರೋದ್ರಿಂದ ನಮ್ಗೆ ಒರಿಜಿನಲ್ ಕೀ ಇಲ್ಲಿ ವರ್ಕ್ ಮಾಡಲಿಲ್ಲ. ಯಾಕೆ? ಫೇಕ್ ಕೀ ಹೋಗಿ ಅಲ್ಲಿ ಕೂತ್ಕೊಂಡ್ಬಿಟ್ಟಿದೆ, ವಾಪಸ್ ಹೊರಗೆ ಬರ್ತಾ ಇಲ್ಲ, ಲಾಕ್ ಆಗಿದೆ. ಕೀ ಹೋಗಿ ಲಾಕ್ ಆಗಿದೆ, ಬಟ್ ಲಾಕ್ ಓಪನ್ ಆಗ್ತಾ ಇಲ್ಲ. Okay, so here we can say this is the enzyme. For this enzyme, the substrate only has to bind. But in the place of substrate, the inhibitor has come, because the inhibitor is similar to the substrate. So the substrate cannot bind with the enzyme, lock cannot be opened. Means this key cannot be inserted into the lock, so lock cannot be opened. Substrate cannot bind with the enzyme, so reaction cannot occur. ಹೌದಾ? ಅಲ್ಲಿ ಆ ಲಾಕ್ಗೆ ಸಫಿಷಿಯೆಂಟ್ ಕಂಪ್ಯಾಟಿಬಲ್ ಆಗುವಂಥ ಕೀ ಸಿಗಲಿಲ್ಲ, ಹಾಗಾಗಿ ಲಾಕ್ ಓಪನ್ ಆಗಲಿಲ್ಲ. ಅದೇ ರೀತಿ ಆ ಎಂಜೈಮಿಗೆ ತಕ್ಕಂಥ ಸಬ್ಸ್ಟ್ರೇಟ್ ಬರಲಿಲ್ಲ, ಬೇರೆ ಅವರ ಬಂದು ಅಕ್ಯುಪೈ ಮಾಡಿದರು, ಹಾಗಾಗಿ ಎಂಜೈಮ್ ಆ್ಯಕ್ಷನ್ ಆಗಲಿಲ್ಲ. ಓಕೆ, so now this is our inhibitor, this is our enzyme, this is the substrate. Okay, so ಈಗ ಹೇಳ್ಬೋದಾ what is inhibitor? Inhibitor is the substance which is having a similar structure like a substrate, binds with the enzyme and inhibits the or suppress the enzyme activity. Okay, a chemical that binds with the enzyme and is similar to the substrate and suppresses the enzyme activity, that is called inhibitor. Okay, and this process is known as inhibition. The process is called inhibition. Okay, this substance that binds with the enzyme, which is similar to the substrate. Substance similar to substrate binds with the enzyme, suppress the enzyme activity, is called inhibitor. And the process where the inhibitor binds to enzyme and suppress the enzyme activity is called inhibition. Okay, so this is how the inhibition works. Okay, so when it exactly similar, when it's almost similar, then it is called competitive inhibition, because this is competing for the space. In the place of original key, this key has come, so this is competing for the space. Okay, so then it is called competitive inhibition. Okay, but we will write the definitions of this. So don't draw this in examination. Okay, just for understanding, I told you, don't draw all this in examination like a key, lock, home, door, etc., etc. Just for understanding. So inhibitor is the substance that is similar to substrate, binds to enzyme and suppress the enzyme activity. Suppress the enzyme activity is called inhibitor. Okay, so competitive inhibition, when the when the inhibitor competes for the active site. When the inhibitor competes for the active site, then it is called competitive inhibition. Competitive inhibition. I will just draw a simple diagram so that you can understand better. Oh, I have an enzyme here. I have an enzyme here. Okay, so this is my enzyme. I have a substrate here. Better I'll write it over. Enzyme here. Okay, I have a substrate here. So this is the active site. This is the active site. Active site. This is the substrate. This is the substrate. Okay. Here, similar to this, we have another compound. We have another molecule. So this site is not that important for the enzyme action, but this site is very important. So this site and this site both must have to take place. I mean, they have to attach each other. Then only the substrate can become product. But in the place of this site of the inhibitor will come and bind over here. Okay, so here there is no modification for that, but this will occupy the active site. As a result, the substrate cannot bind to this, means both are similar in shape, so in this active site of the enzyme, this site of the inhibitor will come and bind, so that this cannot bind, this will bind. As a result, enzyme cannot perform properly. So it is called competitive inhibition, either inhibition or competitive inhibition when it competes for the space. Space means active site. Okay, for example, we have an enzyme. For that, we'll understand better. So succinate will convert into fumarate by the action of an enzyme succinate dehydrogenase. Succinate dehydrogenase. Okay, ಈ ಎಂಜೈಮಿಗೆ ಈ ಸಬ್ಸ್ಟ್ರೇಟ್ ಬೈಂಡ್ ಆದರೆ ಅದು ಫ್ಯುಮೇರೇಟ್ ಆಗಿ ಕನ್ವರ್ಟ್ ಆಗುತ್ತೆ. ಓಕೆ, ಸೊ ಫಾರ್ ಎಕ್ಸಾಂಪಲ್, ಇಫ್ ವಿ ಡ್ರಾ ದಿಸ್ ಸ್ಟ್ರಕ್ಚರ್ ಲೈಕ್ ದಿಸ್, enzyme, this is the substrate here, succinate, this is the succinate dehydrogenase, then this will convert into some product, okay, any structure. This has to bind over this. What happens? There is another chemical called malonate. Which looks like this malonate. So this site will go and bind over this site, As a result, this will bind to this enzyme, will form some other product other than fumarate. But if this will go and bind here, both will act to form fumarate, normal reaction. But abnormally, this will go and bind here, means these two are similar in structure. Succinate, malonate are similar in structure, so that the enzyme action will not be completed. Okay, that's how the competitive inhibition occurs in a living systems. Okay, so this is a brief idea about the various factors affecting the enzyme action. Okay, water, radiation, they don't have much effect on enzyme action, so don't worry about those factors. As of our examination point, even from NEET point of view, these three factors are very much important. Okay, so I think this will complete our chapter biomolecule, right? So if you have any more doubts in the examination, in the class subject, please post your questions in the doubt group, so that we will come to you very soon with the answers. Okay, I think this is the end of biomolecule, right? So till that, we'll come back with the next chapter. So keep learning. Again, I wish you all the best for your further classes and upcoming examination, and I wish that you will come to the classes very soon. Okay, maybe in a couple of months. Till that, keep learning. Thank you.